Alright, hi everybody. Again, hi, rest of the class. We're finishing up meiosis today. So yeah. Couple things going on. We're going to look at this video really quickly. So we're going to look at the video really quickly, have a review of meiosis, and then spend most of class time yapping about how come you can get genetic variation. So here's this video. It's kind of loud. Nope, it's not kind of loud. It's kind of quiet right now.
Watch how the chromosomes move through.
Is that loud enough? Back row, is that loud enough? Do you want louder? Not louder. Louder? No, it's good. Okay, perfect.
Of meiosis. This time-lapse video uses differential interference contrast microscopy to image part of meiosis 1 in a Drosophila spermatocyte. Notice how the chromosomes, which were lined up at the equator in metaphase 1, are pulled to the opposite poles in the cell of anaphase 1. Credit: Conley L. Ryder, Wentworth Center, New York.
Okay, beautiful, right? Okay. Reminders of things. I know, I keep saying the same thing over and over, but it's easy to get it kind of mixed up. So just to make sure we're all on the same page here, meiosis is going to be different than mitosis, but in many ways looks similar. So meiosis is going to go through something called meiosis 1 and meiosis 2. Meiosis 2 is important, but it's not all that exciting. It's basically the same thing as mitosis. Meiosis 1 is where all the weird stuff goes down. So, what we have in meiosis 1, you'll note we've got 3 pairs of chromosomes, right? In meiosis 1, and those chromosomes have synthesized DNA, and so each chromosome is 2 sister chromatids together. So far, so good? Meiosis 1, those homologous chromosomes are going to find each other. They're going to line up at the metaphase plate. They're going to pull apart. Sister chromatids stay attached. You end up then, and then the cell splits. You end up with 2 cells, each that only have 1 copy of each of the homologous pairs. And then you can line those chromosomes up at metaphase plates, pull them apart, and you end up going from 1 cell, you end up going to 4 haploid cells. Instead of 1 diploid cell, 4 haploid cells. Everyone's cool with this? These things in just bigger picture form for your online studying convenience. Alright. This next part is where it gets kind of cool. A little bit. So, what we're looking at in a picture like this is we've got, and I'll draw on the board in a second, we have sister chromatids, right, being held together. Cool. They're held together by a protein called cohesins, but they're being held together. There's a homologous pair, so there's the homologous chromosome, is basically this, it's the chromosome that's the buddy of the first one I'm talking about. The genetic information on it might be slightly different, but it's coding for the same genes. Okay? So far, so good? What happens is one chromosome and the other chromosome of the homologous pair get held tightly together during meiosis 1. Right? Like they're jammed up right next to each other. And I'm going to do it with my fingers here, but then I'll draw it on the board. Those 2 sister chromatids that are right next to each other physically in space can swap genetic material amongst themselves. It's called crossing over, and it looks like this. Make sure see the board. Okay. We've got, sorry, it's weird to do this backwards. Chromosome that's been replicated, right? Sister chromatids. These two are identical. Yeah. It's homologous pair. These are not identical, but they're really similar, right? I should draw these closer together. Okay, so far so good. During meiosis 1, they're smooshed next to each other. And what they can do is, on these two sister chromatids that are next to each other, this one out here is too far away, this one out here is too far away, they're not participating. These ones here, DNA will break. The DNA will break right here, and the DNA will break here, okay? And then you connect the DNA back up, but you connect it crisscross. You basically cross the wires. Do you see that? So then, when this chromosome is pulled away, it's taking this chunk with it. Is everyone okay with that? It's called crossing over, and it's physically, genetic information from one chromosome crosses over to the other one. And now this is incorporated as part of this sister chromatid right here, and it's attached. While we're looking at this, the genetic recombination that happens if you cross over once right here, this piece is the new piece on the chromosome, right? If instead of crossing over right here, you crossed over back. If instead of crossing over right here, you crossed over right here, you have different genetic information. Okay with that? You could cross over any place along here. And even if you just had one crossing over event, you could have a very different set of options. Right? We'll come back to that. So, crossovers can happen. It's going to exchange genetic material between homologous chromosomes. You're not going to exchange genetic material between like chromosome 1 and chromosome 14. No, they're not buddies. It's the homologous chromosomes that are going to line up, and you share information across those two. Questions about that? Okay, it gets cool. It gets very cool. Down, the light comes off. Okay. I need to keep my life organized. I'm not super great at this, but I try. Okay, so, yeah, you have these homologous pairs next to each other. There's going to be a, the non-sister chromatids are going to have a break. The DNA breaks are going to be repaired, and DNA from one non-sister chromosome goes to the other corresponding segment. Great. So this is just the review of what we saw, but now actually with different words. We're here in meiosis, right? In this cell, we've got 2n equals 6. We have 3 pairs of chromosomes. Meiosis 1, we're going to have those homologous chromosome pairs find each other. They're going to do crossing over. They're going to line up along the metaphase plate. And then the homologs are going to separate. And then in meiosis 2, you're going to have these chromosomes line up along metaphase plate, split apart. And now you have unique daughter cells. We're good with this? Great. everything that's fancy and special about, okay, not, the unique parts of meiosis happen in meiosis 1. So, right here, the crossing over, that's a meiosis 1 thing. It happens here during prophase. During prophase 1, actually. That you line up homologous pairs, that's also happening in meiosis 1. That you separate homologs instead of separating sister chromatids, that's also meiosis 1 thing. All the special fancy stuff is meiosis 1. Meiosis 2 looks a lot like mitosis, except you don't have a double set of chromosomes. You only have one set of chromosomes. Yeah? Okay. That, then, is going to enable genetic variation amongst offsprings. This whole meiosis idea, because you're going to end up with a gamete that has half the genetic information of the parent, and you're going to combine it with another gamete that has half the information of a parent. But the combinations and the ways you can do this are huge. So, right, I said this. There are three big things that are going to contribute to genetic variation. They're going to be independent assortment of chromosomes. We'll talk about all these. Crossing over, and then random fertilization. Okay, those are the three big ways. Just to set this all up, the original source of variation amongst DNA is going to be from mutations. So, mutations are going to produce different versions of the gene. So there'll be a gene for something like potato protein. Okay, like, well, I guess there's lots of proteins. It's going to be a gene for one enzyme that breaks down starch in you. Okay, you've got this enzyme. They're in evolutionary time, right? There was some original. This is the code to make the protein to make this thing, right? As this gets replicated, sometimes mistakes get made, and so there's going to be a slightly different version of this. It's still going for the same thing. It's still a protein that's coding. Sorry, it's still information for a protein that's coding for something, but it might be a little bit different. You might have done this if you've ever, I don't know, taken down a recipe over the phone, right? So like you're listening, and they're like, okay, you can put flour, you're going to put this much salt, you can put this much sugar, you can put this much cinnamon, right? You're taking this down. And maybe you aren't paying a ton of attention, and you say that you're supposed to put in a teaspoon of cinnamon rather than a tablespoon of cinnamon, right? Like, your cookies aren't going to be dramatically different, but they are going to taste slightly different. Okay, like, whoo, I like cinnamon, right? Or maybe you're going to switch nutmeg for cinnamon, right? Or maybe you're going to add slightly more sugar, slightly less sugar. Typically it's going to be very, very similar. It's entirely possible that you take this down and they say vanilla and you write cyanide. Like, that could happen, that's a problem, right? But in general, it's just going to be a little bit of a wobble, okay? But then, now you have this recipe that's slightly different, because you say 2 teaspoons vanilla instead of 1 teaspoon vanilla. So you have the recipe that's 2 teaspoons vanilla. When you give that recipe, people might wobble a little bit from what you gave, right? And you're going to have a whole bunch of different variations. So far, so good? All that's to say, the gene, there's a gene for, not really for cookies, but with analogy, there's a gene for cookies, and there are different versions of that cookie. There's a gene for eye color. There's actually several genes for eye color, but there's a gene for there's a gene for earlobe attachment, and there's different versions of that. There's having the earlobe hang free, and there's having the earlobe attached right to the face. Just so you know. There's a gene for if you can do that curl your tongue thing, right? And there's the gene has different version that's like, yeah, no, you can't do that. So far, so good? Okay, so mutations are going to originally create these differences, but mutations are rare and they're often not helpful. Sometimes they are, but they're often not helpful. But there's a whole bunch of different alleles, a whole bunch of different versions of genes. So, what sexual reproduction is going to do is it's going to shuffle those genetic variations. The big way to do this is the independent assortment of chromosomes. So, homologous chromosome pairs are going to line up, and they're going to line up randomly at metaphase 1. And then, in independent assortment, each pair of maternal and paternal homologs are just going to sort it out differently. So online people, right before we started recording, the class each was a pair of homologous chromosomes, and they got to have, like, thank you for screwing me here. They got to have chromosome 1 from dad, chromosome 1 from mom, and they randomly shuffled those. They flipped a coin, and they, based on the coin flip, they picked which one to give to the offspring. It's a flip of a coin, which one is given, right? And because each chromosome has a random, of each homolog, which one is given is random, that means that the possible combinations are huge. So it's 2 raised to the nth power with ending the haploid number. So if there is 10 of you, it's 2 to the 10th is the possible combinations. For humans with 23, that's just over 8 million possible combinations of just no crossing over, no mutations, just which ones get given. Does that make sense? Are we seeing where this goes? Right? Because you could have one possible combination where you have the, I'm trying to make a gamete, and for this gamete making, I'm going to pull my maternal chromosome 1, my maternal chromosome 2, 3, 4, all the way down. Or I pull the chromosomes I got from my dad. I pull my paternal chromosome 1, 2, 3, all the way down. Or I have all of them except 1. Or I have all of them except 2. Right? Do we see this? So many possible combinations. So, here's the picture here. It's called independent assortment because it's saying this possibility, how these chromosomes line up here, they line independently. So this is an option, right, where all the blue chromosomes are on one side, all the ones that this organism got from their dad. But they don't have to be together. They could, possibility 2 is just as likely, right? These chromosomes don't remember which one came from their father, came from the mother. They just line up independent of each other. So far, so good? So independent assortment says the homologous chromosomes will arrange themselves in the metaphase plate, but they'll do it independent of each other. Then when you pull the homologs apart, you have equally possible outcomes. And then, when you look at the daughter cells, you have 4 equally likely combinations. And each of these combinations are different from the parent, right? And they're haploid. But they're also different from each other. These ones have all blue, these ones have all red, these ones have 1 of each, but the long one's blue, these ones have 1 of each, but the long one's red. Do we see this? This is, again, before mutations, before random crossing over, there are 8 million possible combinations of these things. With 2 chromosome pairs, you have these 4 options. With 23 chromosomal pairs, you have more than 8 million options. Right? Okay. It gets more exciting. I know, right? Like, edge of your seat, people. Yeah. Sorry, I get excited and I don't know where to go from things. Okay. You have those, just me by myself, for the possible gamete combinations I can make. I've got more than 8 million. I can make 4 possible offspring. That was before crossing over. If you look at crossing over, you suddenly dramatically increase the options of DNA inherited. So crossing over is going to make it so the DNA from two parents can combine into one chromosome. So it's not super. So crossing over happens for each chromosome during meiosis between 1 and 3 times typically. It's odd for no crossing over to happen. It's also odd for like 5 crossover events to happen. But it's going to make things mix things up dramatically. So in this example here, you have a cell which for simplicity's sake only has 1 pair of chromosomes, right? It has 1 pair of chromosomes. During meiosis 1, they cross over, and then so this 1 pair of chromosomes, it's had some crossing over events. It splits into these daughter cells. Yeah, you have one daughter cell that has the chromosome that looks just like the paternal chromosome. You have one daughter cell that looks just like the maternal chromosome. But you have these 2 daughter cells that don't look like either of those. Do you see this? They don't look like either of those, but they also don't look like each other. Right? And it's not like there's magically different DNA, right? This is DNA from dad, this is DNA from mom. But this DNA from mom and DNA from dad are now on the same chromosome. Are we okay with this? You can have so many different options. This, if we were going back to the little activity at the beginning, this was then, instead of having where you have one hand A and one hand B and you flip a coin, which one do you get? Before you do that, you rearrange things a little bit. You're like, okay, this one's A, this one's B, but you know what? I'm putting some of B onto A. and some of A onto B. And so then, when you pick, it's A. It's like, well, actually, it could be A or it could be 90% A and 10% B. Do you see this? Okay. Questions? Great. And so right here, we're looking at a crossing over event. Just see you've got this chromosome here being held there. It legitimately crisscrosses. Oh, sorry. It forms a chiasm, just Greek for X. Crisscrosses and then pulls back apart, but you've switched. And I won't draw on the board because it's hard for online people. If you cross once and you cross far out, right, you change the little end of DNA. If you cross way closer up in here, right, you change a lot more of DNA. But you can cross more than once. So you could have a crisscross way out the end, and another one right here. So you just shift a little bit. You could have a crisscross and then another crisscross event way over here. Right? You could have any combination of this. Yeah? Okay. Here's our exciting video. Online people, it's in your on the PowerPoint notes. This is, yeah. It's everything we just said, but yeah, it's exciting.
Producing organisms, 3 processes lead to most genetic variation. Independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
So far, so good? Okay. So, where we're at, we've had independent assortment, right? Where those homologous pairs can line up in any order along the metaphase plate. So again, not counting mutations, not counting crossing over, just by which order I put my homologous pairs in, I can have 8.4 million different gamete options, me personally, right? Now, if I've got 8.4 million options, right, and I pair that gamete with my mate, right, who also has 8.4 million options, right, there's a total possibility for the zygote, again, not counting crossing over, this is before crossing over, of about 70 trillion different combinations, okay? 70 trillion is what is defined as a very big number, right, like a lot, okay? The 70 trillion even before crossing over. And then if you count crossing over, which can happen anywhere between 0 and, I guess, however many times, but typically it's happening about twice per chromosome. You have so many possible combinations. We don't even know how many possible combinations exist. Do people see this? People okay? The take-home message from this is that you personally are genetically unique. The genetic information that you have has never before, with any organism, that combination has never existed before. Right? So, like, feel special. Yeah. Okay? So, you might look very similar to your great Uncle Lester. It's adorable, right? But you still have a completely different set of genetic information than anybody else. We're okay with this? It's a lot. So yeah, you have independent assortment that makes a unique gamete. You have fusion of 2 different gametes that come together to have 70 trillion different combinations possible. The crossing over that happens is going to provide even more variation. So if you're an organism that uses sexual reproduction, you have a huge variety of possible combinations. So far, so good? You've heard of, like, survival of the fittest? Yes? Okay. The idea is natural selection favors genetic variations that work well. So sexual reproduction is going to contribute to what's called genetic variation in population. So, as an example, because I'm stuck on the potato concept from whenever, potatoes can reproduce sexually, they can have seeds, right? But the normal way to grow potatoes is to get a potato, cut it up into little bits, and grow those potatoes, right? To have clones of these potatoes. Okay. Has anybody heard of the Great Irish Potato Famine?
Yes.
Was it a problem? Yes. Millions of people starved? Okay. It's a problem. If you haven't heard of that, you're about to hear it. 1800s in Ireland. If you're just the average kind of person, what you're trying to do is you're trying to not starve, right? So you've got probably a very small plot of land. It's probably not super great land, but you can grow potatoes. And these potatoes that you're growing, originally from like Peru, that's cool, they're here, they're great. They grow really, really well. They grow really well and you can eat them, and potatoes are delicious, and they grow in your little tiny plot, and they're great. And the way you're growing these, because this is how potatoes work well, is you take your potato, you cut it up, you plant all those chunks, and you have potatoes that potato plant 1 and potato plant 2 are clones of each other. Yeah? And potato plant 1 and potato plant 2, they're clones of each other. And your next door neighbor buddy has the same ones, right? You all have basically the same potatoes. Okay? Yours are special because you take good care of them and they're beautiful. But genetically they're all the same. Right? Okay. They all have the same strengths, right? They're delicious, especially tasty if boiled or whatever, right? Okay. Problem is, if there's, say, a disease that your potato plant number 1 is susceptible to, this little disease comes in and it kills potato plant number 1. What's potato plant number 2's likelihood of being better at fighting the disease than potato 1? Not happening, right? If this disease takes out potato plant number 1, it's just tearing through the whole thing, right? The potato blight just wiped out the Irish potato crop, like wiped it out. In large part because these potatoes all had the same weaknesses. Okay? If you don't have sexual reproduction, for complicated organisms, you expect that the organisms are going to have very similar strengths, but very similar weaknesses, which can be great until suddenly it isn't. And then it's really, really bad. So animals that reproduce asexually are really, really rare. Things like bacteria will produce asexually, but bacteria have an edge on this because they just mutate all the time. Like, mutations happen in any organism's DNA, but like, you are very paranoid and picky about not having lots of mutations happen in you, right? Bacteria, sure, just put stuff together, whatever, right? They change really, really quickly. Complex organisms don't usually do this, and so, yeah, the genetic variation that's getting picked up is a huge part of this genetic variation is going to become because sexual reproduction. We're cool with this? There are some animals that can reproduce sexually as well as asexually. Again, some plants can do sexual and asexual, right? You may have heard of some like lizards that can do this. No? Yes? On Friday, I need to show you asexually reproducing lizards. I don't know, would this make your Friday better or worse? Better? Okay. Because I realize in some ways that could be a threat. Like, I will show you a video of, I don't know. Off the top of my head, I don't have a good video for this, just off the top of my head, so I won't look for it, but on Friday we'll see it. It might be fish. Lizards are probably the best ones, though. But they can do either. Animals that, like, straight up just reproduce asexually, super, super rare. There is, your book talks about, I'm assuming you're reading your book, like, religiously. I'm going to go with that assumption, nobody correct me, but let's assume. Your book talks about a little buddy called a rotifer. It's kind of adorable. That reproduces asexually. And that's bizarre. Like, it's bizarre to an animal that does that. the studies, when this was studied, it looked like, and your book still talks about, it looks like the little road dipper buddy is like hanging around in the environment, happy to reproduce asexually, but to get genetic variation, it just like finds DNA and just like boop into it. So it's like, doing its thing in this whole water, right? Because it stays in moist things, and finds a little chunk of DNA. It's like, ooh, I like that, right? There's currently, I made these slides like a week ago, but I just was reading more, and it's entirely possible that there might have been some contamination problems with that experiment. So I'm not actually positive now. There's some skepticism on that experiment, but it's entirely possible these little buddies are reproducing asexually, but they're getting genetic variation because they're just like stealing DNA from other stuff. You can't do that, right? So like, you can eat a piece of celery, nom, 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 delicious, but you can't like incorporate celery DNA into your DNA to get variation. You don't do that. So if you want genetic variation to happen in the species, sexual reproduction is kind of the way to go. Questions? Comfortable with that? Okay, in the last, it should be like 8 minutes. 7. Okay. Not good at like timing things in general. So when I'm remotely close to time, it makes me very happy. I'm like, ha. But like painfully bad at, like, we'll have dinner and they're doing something, like, I have no idea what time it is. It could be 7, it could be 9, like, I don't know. It could be any time. Okay. 5 questions here, and I realize we're losing people fast. Like, not dead, but like people are going home or being sick or other things. Glad you all are still here. Try to find a buddy or a set of buddies. Do you feel lonely? Yeah. Ellie and Dex are nice. You'll be buddies, right? Okay. Find a buddy, ask them a question, see if they're right. They'll ask you a question, etc., etc. Great. I'm gonna like nosily pop around. Oh, you two together? You three want to sit together then? Right? Like, you're on. You're so ready. Was that a long enough time to at least get through a couple? Sweet. Any ones that are super complicated? From what I heard, and I was trying to sneakily listen to people, it sounds pretty good. It sounds like the independent assortment one might be the trickiest. Is that true? I think I heard, and sorry, it's challenging to like creepily listen to everybody talking quietly. I think Dax gave a really good answer for it. Does anybody want to take a shot at it? Yeah, go for it.
Okay, so the independent assortment would be like, just like the whole different bunch of combinations of different chromosomes together.
Yeah. Yeah. Is everyone cool with that? And sorry, I'm going to repeat, like, it's a beautiful explanation. I'm going to repeat because the online people, it's easier to hear. Dax was saying, so in pet assortment, you've got these homologous chromosomes, and they can line up any way. So you have a whole bunch of different options for which version of the chromosome is making it into the gamete, right? The rest of them, pretty good. People okay? Great. That's what we have. A couple things to remember. You have your lab exam. Yes? When is that due by?
Friday.
Oh, you guys are so good. Okay, yes, it's due by Friday. Next week, on Wednesday, you're turning in lab, you're turning in your paper draft, right? Your exams are graded. I can't show them to you quite yet because there's one person who, for non-interesting medical reasons that the whole class doesn't even know about, can't take it right now. But they're up, and people did really pretty well, so that's good. I still am impressed with the impressed and kind of horrified with one person who did it in 6 minutes. I'm like, oh gosh, it makes me deeply physically uncomfortable that they did that fast. But cool, more power to you. But most people took longer than that, and that's perhaps wise. But yeah, I think people did a good job. So yeah, you'll see that soon.